I'll start by introducing our first speaker. So first up, we have Rob Finnegan. Rob is a medical physicist, cancer researcher, and currently works at Northern Sydney Cancer Centre. And today he'll be talking about unlocking medical image with PlatiPy, an open source software that he developed. Over to you, Rob.
So let's just get started. So I live and work in the lands of the Camaragal people of the Eora Nation, and I'd just like to acknowledge their traditional ownership of country and pay my respects to elders past, present, and future. For those of you who don't know, the Camaragal people live in the lower North Shore of the Sydney Basin, and so that's where I'm joining you from today. And I'd also really like to thank everyone who's been involved with the various research projects from which this software has been inspired and developed to solve problems in. And in particular, Phil Clapp, who's a senior radiotherapy computer scientist who's been instrumental in helping clean up my terrible spaghetti code. So I thought I'd start with a bit about Northern Sydney Cancer Centre and about radiation therapy in general, given the audience and the focus of this conference is predominantly around MRI. I thought it couldn't hurt to give a bit of background and context. So this is where I work, Royal North Shore Hospital, and Northern Sydney Cancer Centre is in this back building with the green bars on it. So we are a large tertiary institution and we treat around 1,200 patients every year with radiation therapy. There's a large multidisciplinary team, over 100 staff work in the radiotherapy department, and we have close collaborations with academia and industry as well. We're quite a progressive department, and I think we would consider ourselves an early adopter of technology. This includes hardware solutions like treatment and imaging technology, but also a lot of software tools. So let's go on to radiation therapy and discuss the patient journey. So once a patient's diagnosed with cancer and prescribed radiotherapy, generally the first step is that they'll have some imaging done. Often this is CT imaging. And the reason we use CT imaging a lot in radiation therapy is because we need the electron density information in order to calculate dose. So while other imaging modalities are used very commonly, still there's a reliance on CT. Doctors and other healthcare professionals will then delineate different parts of the image so that we can develop a 3D model of the patient anatomy, and we can use this model to design an optimal treatment regime for that patient. This is called the planning process in general, where we design radiation beams and optimize the way that they pass through the anatomy so that we can deliver a high radiation dose to the target, which is often the tumor or the organ the tumor is in, like the prostate in this example, and try to spare healthy tissue as much as possible around that. And then the patient's treated on a linear accelerator or other treatment machine. We use Varian true beams, and I've shown a picture there. And typically patients will receive anywhere from 1 to 30 fractions of their treatment. So how is MRI used in radiation therapy? It's used in a lot of different ways. Probably the most common uses is complementary imaging to the CT that we acquire for radiotherapy planning. So I've shown here a case from our department where a patient was treated for medulloblastoma, brain cancer, and MRI is acquired because it, of course, has excellent soft tissue contrast. We use multiparametric MRI a lot in the treatment and management of prostate cancer patients as well. Increasingly, frameworks have been established to facilitate MRI-only planning so that we don't need to acquire a CT. And in our clinic, we treat gynecological conditions with brachytherapy and only use MRI in this case. If you saw David Waddington's talk yesterday, you might also have seen some new integrated systems that combine an MRI scanner with a linear accelerator. There's currently 2 commercial models available, the Electa Unity and the ViewRay Meridian. They're pretty similar. The ViewRay Meridian has a much lower field strength, about 0.35 Tesla. And they have a rotating gantry, so the linear accelerator can deliver radiation from multiple directions. So why do we care about software development at all? Well, medical physicists wear a lot of different hats in the clinic, and I'm not going to go through all of these different reasons, but suffice to say that radiotherapy is very complex, and we often want to make sure that what we're doing is the best we can do. And so, given there's so many software systems involved, developing software underpins a lot of tasks that we do, and also helps us a lot in research. So why open source software then? I've sort of identified a few key areas. It gives you access to the cutting edge technology, which is generally released before commercial products are developed. It's also a great way to collaborate and strengthen professional networks in the field. Open source software, by its very nature, means you can take it and do what you want with it, adapt it to your specific use case. And last but definitely not least, it's really fun. Like many of you, I really enjoy developing open source software and sharing ideas in this way. PlatiPy really depends on a large body of existing open source software that goes back decades. And in this respect, we're standing on the shoulders of giants. So I feel like releasing software as open source is a great way to add another brick in the wall. So what is PlatiPy? You can see the acronym for it down there. It's basically a library of tools and functions to try to facilitate analysis and processing of imaging. When I first started in this field, I was really captivated by the beautiful imaging. And after working for a while, I started to realize that at the heart of it, everything is an image, not only the CT scans and MRI and PET that we're used to thinking of images, but things like dose distributions and scalar maps that correspond to other functional information, contours, delineations of organs at risk and planning volumes, and a whole lot more. And so given everything can be represented in this sort of voxelized array, common tools to integrate between those prove to be very powerful. And so that's how PlatiPy was born. Who's PlatiPy for? Everyone. I guess simply, PlatiPy has a lot of ready-to-use tools. So if you're just looking to pick something up and integrate it into your workflow, PlatiPy could be very useful for that. But being open source, you can also pick apart the different components of the algorithms, maybe put them into frameworks that you're developing for research or clinical use. And a big part of PlatiPy revolves around advanced visualization tools, and I'm going to be touching on that throughout the talk. PlatiPy is available on pip as well as GitHub, so you can just install it from your command line interface. And to use PlatiPy, you can access the functions available on the command line interface. So for example, there's a bunch of segmentation tools, which you could run like that. You can also import PlatiPy functions into your Python code and use them directly. And for advanced power users, PlatiPy also comes with a really powerful server client framework so that you can run different tools on a server and send DICOM requests to process your data automatically. I've broken down the features that are available in PlatiPy into some different categories, and I thought I'd just go through some examples of each of them to give you a taste of what's available. For more information, you can check out the paper that we recently published in JOS, or even better would be to check out the tool on PlatiPy, where we've got a bunch of Google Colab notebooks, and we're always trying to add more so that you can actually just jump straight in and see how you might use them. So let's start with conversion. Love it or hate it, DICOM is the standard for medical imaging. It's a very complex data format and made even more complex by the fact that there's often interrelationships between different files. So in radiotherapy, of course, we have the imaging, but linked to each of these images, we might have sets of RT structures, which are segmentations and dose grids. These can be very challenging to work with, particularly in research when you're trying to analyze large data sets of retrospective patients. And so converting to more easy to use 3D formats, lots of which actually came out of MRI, makes a lot of sense. And for this, we've got possibly my favorite tool, the DICOM crawler. With just a simple command, you can iteratively search through an entire directory and convert all of the DICOMs in whatever strange sort of organizational format they're in into easy to use 3D formats in nice tidy directories. And this is part of a larger ongoing project, the Australian Cancer Data Network. If you're like me, you probably find yourself writing the same 4-line functions over and over again. And it's really important to try to reduce this boilerplate code, in my opinion, because it opens you up to errors. And so we're just trying to add to PlatiPy simple functions, even things like computing centers of mass and bounding boxes, just so you can quickly and easily access the information you're after. Image projection is another tool that's very useful. It gives you a nice way to visualize an image, and it can also be used to transform between 2D slices and 3D volumes, which you might use for training models, for example. And being able to view 3D volumes at different angles in radiotherapy makes a lot of sense because often we're treating from different angles. So projecting along an arbitrary axis is a useful function here. Being Python code, PlatiPy also integrates well with a lot of existing Python libraries like PyRadiomics and SimpleITK, so that you can integrate PlatiPy into your existing frameworks that might use these libraries. Image registration is incredibly important in many fields in medical image analysis, including radiotherapy, where it could be used to develop atlases of typical anatomy or to propagate contours from a planning CT to a CT taken during treatment, for example. And PlatiPy has some really great functionality to very quickly and easily register 2 images. So with just 1 line of code, you can get started and you have full access to the different parameters that are involved in setting up registration. So this is just an example of a linear registration between 2 different patients. It's also got some great tools for deformable image registration. And again, with just 1 line of code, you can get started with that. Image registration is, of course, sometimes seen as quite a black box. And so we thought it was really important to build in some tools so that you can understand what's happening during the registration process. So you can see here on the left, the deformation vector field, describing how one image has been mapped to another during this process. In the middle is the magnitude of that. So you can see parts of the image that undergo larger deformations. And on the right is a Jacobian determinant, which is a mathematical way of calculating how much a small subvolume either expands or shrinks during the registration process. So you can see that the lungs have had to expand quite a lot during this registration, for example. Like registration, segmentation also underpins a lot of analysis that we do. So extracting out regions of interest, developing models so that we can predict the anatomy on a new patient. Out of the box, it comes with some basic auto-contouring models, which we can use to delineate things that have quite distinct intensity profiles. But it also comes with multi-atlas auto-contouring, where if you have a library of patients where the images have already been segmented, then in just a few lines of code, you can get started with developing a model to delineate new unseen images. Many of you have probably heard of NNUNet. It's one of the state-of-the-art frameworks for deep learning-based auto-contouring. Rather than reinvent the wheel, we've also integrated NNUNet within PlatiPy to make it easier to run some segmentation models, which you can download from the web or train yourself. If you're anything like me, once you actually perform a segmentation, you want to know if it worked or not. And so visualization is key to that, but more than visualization is probably getting some numbers. So PlatiPy provides simple tools to calculate different metrics, geometric measures of similarity between 2 segmentations. So one could be manual and one could be automatic, or they could be 2 different models that you're trying to compare. And there's a whole range of metrics that PlatiPy gives access to, including some that are relatively new, like the surface dice or added path length. PlatiPy also provides simple ways to automatically generate reports, which can make it easy to facilitate retrospective analysis or analysis of large data sets. I thought I would put in some information about how visualization is key to understanding your data. And I've used an example of the dose distribution that we would deliver to a breast cancer patient. So you can see that the dose in the breast that we're treating is quite high and it sort of falls off as we move away. But there's different ways, of course, to visualize this scalar field. And the different ways to visualize it can give you lots of complementary information. So you could use different color maps that help you identify regions of high gradient or where there's particular hotspots in your scalar field. You could look at the low dose wash, which represents where radiation is, which represents the lower amount of radiation to the healthy tissue. And you can combine this with the image projection functionality to get a nice 3D representation. Combining this with segmentations, you can start to extract out quantitative metrics. I won't go through these because they're radiotherapy specific, but I'm sure you can appreciate that you could extract out radiomic metrics and things like that in this way. So hopefully you've seen how visualization is sort of underpins all of the different functionality in PlatiPy. And as well as just the simple static images, there's some really nice animation tools. So you can see here rotating around an image or looking at the magnitude of motion within the heart for a 4D scan, or simply slicing through a 3D volume. So this is one single scan of a patient, and hopefully it gives you an appreciation of just how rich these data are. So as well as these figures, actually every figure in this presentation was made with PlatiPy, which is quite cool, I think. So just to finish up, I thought I would give you an example of a use case for PlatiPy out in the real world. And I would consider this a sort of crown in the PlatiPy world. So just to give you a bit of context, a lot of my research has revolved around thoracic radiotherapy, which is broadly indicated for a wide range of clinical sites. In Australia, tens of thousands of patients every year are treated with thoracic radiotherapy. And part of the problem when they're treated with radiotherapy that goes through the chest is that part of the radiation can go through the heart. Currently, the models for how this radiation affects the heart are very limited. You can see the confidence intervals in this risk model here are extremely wide. And part of the problem why there's so much uncertainty in the risk due to radiation exposure of the heart is because the current clinical standard is just to treat the heart as one single structure. But as we know, the heart is comprised of many different functional subunits like the chambers, valves, conduction nodes, and so on. And each of these might respond differently to radiation. So how are we going to get better risk models? Well, we need to analyze a lot of retrospective data, extract out dose information and relate that to risk, but that requires automatic contouring to define these heart substructures. We developed what we describe as a hybrid model that combines deep learning, multi-atlas, and geometric segmentation tools. And all of this is publicly available in PlatiPy. You can run this segmentation model with just one line of code. And so far, I should probably update this, but it's probably close to about 30,000 CT scans that have been analyzed with this tool around the world, not just by our research group, but by many. And we're really proud of that achievement. There's lots of cool things on the way. Something I'm really interested in is generating realistic deformations so that we can try to optimize the robustness of radiotherapy. My colleague Phil Clapp's been working on some really interesting deep learning functionality to use probabilistic units in our segmentation tools. And as you've heard, cardiac modeling is a big focus of PlatiPy. So I'll finish up there. Thank you very much for your attention. If you're keen to get involved, check out the GitHub, and I'm happy to answer any questions.